Welcome to our journey into evolutionary biology. Today, we are going to explore one of the most fascinating tools scientists use to understand the history of life: molecular clocks. We will discover how tiny molecules inside your cells act as biological time machines, allowing us to trace our ancestry back millions and even billions of years. Now that we know we will be looking at biological time machines, let us understand the basic concept behind them. Imagine a secret family recipe for cookies, passed down for centuries. Every generation, someone might accidentally swap an ingredient, but the core recipe remains. If you compare your recipe with a distant cousin's, counting the differences tells you exactly how long ago your family separated. In evolutionary biology, we do the exact same thing using genetic and protein sequences. By counting the tiny copying errors, or mutations, that accumulate over time, we can trace the family tree of all life on Earth. This is especially crucial because the fossil record is often incomplete. Building on this idea of a steady accumulation of changes, we must ask, which molecules make the best clocks? In the scientific world, we look for highly conserved molecules. These are sequences of DNA, RNA, or proteins that have remained remarkably unchanged throughout history. Why do they barely change? Because they perform a function so critical to life that any major mutation would instantly kill the organism. Since they only tolerate very slight, harmless changes over long periods, they tick slowly and steadily. This makes them perfect molecular clocks, allowing us to calculate exactly when two different species shared their last common ancestor. Now that we understand what conserved molecules are, let us look at our first specific example: cytochrome C. Think of this protein as a crucial gear in the engine of a car. Every single organism that breathes oxygen, from a tiny yeast cell to a massive blue whale, relies on this exact same protein to generate energy in its mitochondria. Because it is so incredibly important for survival, any major changes to its structure would be deadly. Therefore, it evolves very slowly over millions of years. This makes cytochrome C absolutely perfect for comparing organisms that share a relatively recent common ancestor, such as humans, chimpanzees, and dogs. While cytochrome C is great for recent history, what if we want to look even further back in time? For this deep time travel, cytochrome C changes a bit too fast. We need a molecule that is even more fundamental. This is where ribosomal RNA, or rRNA, comes into the picture. Ribosomal RNA forms the core physical structure of the ribosome, the microscopic factory inside every cell that builds all other proteins. Because every living thing on Earth needs ribosomes to survive, the rRNA sequence has barely changed over billions of years. This extreme stability makes it the ultimate deep time molecular clock, allowing scientists to separate the massive domains of bacteria, archaea, and eukarya. Having met our two biological timekeepers, we need a mathematical way to link their mutations to the passage of time. The fundamental idea is that mutations accumulate at a relatively constant rate. We express this relationship using a simple linear equation: D equals 2 times R times T. In this equation, the symbol D represents the total proportion of genetic or amino acid differences between the two species. The symbol R represents the constant rate of mutation, usually measured in substitutions per site per year. Finally, the symbol T represents the time that has passed since the two species split. If the mutation rate R increases, the total divergence D will also increase for the same amount of time. Before we dive into why this formula works, let us see it in action with a concrete example using cytochrome C. Suppose we compare the protein sequence of a human and a horse and find they differ by 12%. This means D is 0.12. Scientific studies establish the mutation rate R as 0.00000001. We are asked to find the time, T. We start by rearranging our formula to isolate T, giving us D divided by 2R. We then substitute our given values into the numerator and denominator. Carefully dividing these numbers gives us 60 million years. This means humans and horses shared a common ancestor approximately 60 million years ago, which perfectly matches the fossil record. Now that we have successfully used this formula, let us understand where it comes from and why we multiply by 2. We are setting out to prove the logic behind the molecular clock equation. Imagine the exact moment two species split from their last common ancestor. We call this time 0. From this moment onward, the first lineage begins accumulating mutations independently at a constant rate, R, over a time period, T. Therefore, the total mutations in this first branch is simply R times T. Simultaneously, the second lineage is also evolving and accumulating its own unique mutations at that exact same rate, R, for the exact same amount of time, T. Continuing our derivation, we look at the combined picture. The second lineage has also accumulated mutations equal to R times T. When we compare the modern DNA of these two species, the total genetic divergence, D, is the sum of the mutations that happened in both independent branches. Mathematically, we add the mutations from lineage 1, which is R times T, to the mutations from lineage 2, which is also R times T. Combining these terms gives us our final result. D equals 2 times R times T. The intuition here is simple but powerful. The factor of 2 accounts for the fact that both species were mutating independently after they separated. Having proven our main equation, let us apply it to our deep timekeeper, ribosomal RNA. Scientists are studying two ancient lineages of bacteria that diverged exactly 1.5 billion years ago. When comparing their RNA sequences, they find a genetic divergence of 6%, meaning D is 0.06. We are asked to find the mutation rate, R. We rearrange our standard formula to solve for R, which equals D divided by 2T. We substitute our known divergence into the numerator and our known time into the denominator. The denominator becomes 3 billion. Dividing these gives a final rate of 0.00000000002. This incredibly small number proves physically how slowly RNA mutates. Let us briefly look at the mathematical reasoning behind the rearrangement we just used for ribosomal RNA. We are establishing how to isolate the mutation rate before plugging in any massive numbers. We start with our proven molecular clock equation, D equals 2 times R times T. In this scenario, geological fossil evidence gives us the time, T, and modern DNA sequencing gives us the divergence, D. Our goal is to find the unknown mutation rate, R, which tells us how fast this specific molecule ticks. Setting up the algebra carefully ensures we do not make mistakes. Continuing our derivation, we perform the final algebraic step. To isolate the mutation rate r, we must divide both sides of the equation by the term 2t. This cancels out the time variables on the right side, leaving r completely by itself. This gives us our final rearranged formula. R equals d divided by 2t. This makes logical sense because the total genetic differences, d, must be distributed evenly across the total combined time both branches have been evolving independently. This simple rearrangement is the key to unlocking deep evolutionary time. Now that we have calculated these rates, let us visualize how they build the tree of life. Here, you are looking at a phylogenetic tree. The horizontal axis represents time, moving from the distant past on the left to the present day on the right. Notice the deep branch point on the far left. This represents a split billions of years ago, separating bacteria from other life forms. As the label shows, we must use the incredibly slow-ticking ribosomal RNA to map this ancient intersection. Moving to the right, we see a much more recent split, just 60 million years ago, branching into humans and horses. Here, the diagram shows we use the faster-ticking Cytochrome C. This visual perfectly illustrates how different molecular clocks map different depths of evolutionary history. We have covered the full topic, so let us reinforce the most important ideas. First, highly conserved molecules act as biological time machines, allowing us to read the history of life written directly in our cells. Second, we learned that different molecules tick at different speeds. Cytochrome C is perfect for tracing the evolutionary relationships of animals over millions of years, while ribosomal RNA is an incredibly slow-ticking clock that helps us map the deepest branches of life over billions of years. Finally, by applying the molecular clock formula, we can translate tiny genetic differences into exact timelines. You now understand how all life on Earth is beautifully and mathematically connected.